We will now discuss HIV human immunodeficiency virus in more details. So virions is similar to all retroviruses, envelope virus, which has conical capsid, which resembles bullet shapes. And that's very typical shape for HIV virions. Genome is, of course, linear, single-stranded RNA with positive sense, and as all retroviruses, HIV packages two RNAs. And as a primer for reverse transcription, it packages tRNA for lysine. Each retrovirus packages particular tRNA, which it uses as a primer for reverse transcription. Genes, it contains a typical GAC, Pol, and N genes. However, in addition, HIV has 6 regulatory genes which play a very important role not only in transcription but also in defense against immune responses. And we will go to them in more detail later. It's VIF, VPU, VPR, TAT, and NF. Now, HIV, as we discussed previously, is lentivirus, which means it has very slow progression to diseases. And there are two types, HIV-1 and HIV-2. Most prevalent is HIV-1, while HIV-2 is predominantly found in Africa. And now, the lentiviruses, as you recall, are SIV, FIV, Isnamidi virus, also caprine arthritis encephalitis virus. So there are several species-specific lentiviruses. So let's go a little bit into the history. HIV virus was discovered in 1983 as a causative agent of AIDS. In 1981, the new disease, very unknown immune deficiency, has been detected in the group of the homosexual male in San Francisco. And later on, it was extended to finding in New York. So at the beginning, it was thought that it was just gay disease. However, later on, it was discovered in heterosexual couples, also in children. So there are still some misconceptions which claim that HIV infect only homosexual. It's not true. HIV actually worldwide, the prevalence of the virus in male and female is approximately 50-50. It's only in US when the males are more affected. So once the disease was discovered and described, many laboratories try to identify the causative agent, and it happened for the first time in 1983. And the discovery of virus was made in the Pasteur Institute in Paris by Jean-Claude Sherman on the left, François Barré-Sinoussi, and the head of department, and they did discover the virus was Luc Montagnier. The discovery led to the Nobel Prize for Medicine in 2008, and sadly, the Nobel Prize was awarded to Barré-Sinoussi and Montagnier because she was the first author on the paper and he was the last one, even though the major role in discovery was played by Jean-Claude Sherman because it was his laboratory where François Barré worked, and he was a leading force behind the discovery. Sometimes things like that happen in science. Now, some major facts about HIV. HIV/AIDS represent one of the major global pandemic. Nowadays, it's estimated that 38 million people live with HIV, and most affected area is Africa. Up to now, more than 30 million people died of AIDS. And the major problem with HIV infection is that the virus infects the cells of immune system and kills them. And the infection results particularly depletion of CD4 positive T cells, which are the major target of the virus. And that, as you recall, CD4 are central for immune responses because they also help to CD8 cells, B cells, macrophages. So the depletion of CD4 cells renders the host immune incompetent. There is acquired immune deficiency. So a result of those immune deficiency statuses are opportunistic infections, which are the pathogens which normally do not cause major pathogenesis. However, when immune system cannot fight them, they may trigger a serious condition. Now, transmission, HIV is transmitted predominantly through sexual contact or blood exchange. And during sexual contact, HIV crosses mucosa, and this is facilitated by abrasion, inflammation, or interaction with dendritic cells, which reside in those areas. Um, Another distinguishing characteristic is loss of immune competence, respectively, not another is a major one. Okay, so keep in mind the major characteristic of HIV infection is marked depletion of CD4 positive T lymphocytes. HIV also infect macrophages because they express CD4 receptor as well and chemokine co-receptors. However, macrophages are not killed. Their functions are altered, but they are not killed. So major depletion is noted in the CD4 positive T lymphocyte population. Now, once virus impact a person's disease usually progress in three phases. First is acute infection, which happen right after the infection or respectively transmission of the virus. And this acute infection can last up to several weeks. In some cases, there are signs. In some cases, there are no signs. Acute infection is followed by clinically latent phase, and if untreated, eventually it can progress to terminal stage, which is AIDS, acquired immune deficiency syndrome. Now, acute phase, as I mentioned, sometimes there are some syndromes, flu-like syndromes. However, in many cases, there are no syndromes, so person may not even know that he or she is infected. And as you can see, during first phase here, the virus is, it's not labeled here, but virus replicates. However, when CD8 are generated specific, they curb down CD4. But after this primary infection, CD4 start to decline. And what is a major characteristic and what precedes the pathogenic course is infection of gut-associated lymphoid tissue. Because during primary infection, there is major significant depletion of CD4 T cell in this area, and that sets the course of infection. Now, the viral titer drops usually due to activation of T lymphocyte, as I mentioned, and virus replicates, but in very low levels. So during that clinical latency, virus still persists in lymph nodes because during that first generation of immune responsive, immune system was not able to eliminate the virus. So virus is still there, and basically it continuously replicates to low level. And this causes gradual depletion of CD4 also in periphery. And if there is no treatment, eventually, as I mentioned, this progresses to AIDS, where virus start to replicate and you can detect very high level of HIV. So high level of virus you can detect during primary infection and AIDS stage. So its sign is depletion of CD4, the level of CD4 drop below 200 per microliter of the blood. There is chronic fever, sweat, diarrhea, opportunistic infection, neurological manifestation, also neuromuscular syndrome, kidney failure, opportunistic infections such as Kaposi sarcoma. And disease can actually progress differently in different people. There are several courses. There are so-called rapid progressors, slow progressors, and long-term non-progressors. Rapid progressors develop late-stage symptoms without treatment in 2 to 3 years, and approximately 10 to 15% of infected people are rapid progressors. Slow progressors develop late-stage symptom in 8 to 10, sometimes 15 years, and 70 to 80% of cases manifest slow progression. And there are some group of people, approximately 5%, which are long-term non-progressor. They show no decline in CD4 T cell in spite of fact that virus replicates. All this development, all these three courses are referring to patient infected, those who are untreated. And we will talk about that a little bit later. So what determine long-term non-progressors? It is still not completely clear. It can be virus, which is different. It can be stage of immune system and so on. Now, as I mentioned, all those courses of infection refer to patients who are not on treatment. Nowadays, we have very powerful antiviral drugs, treatments, which can control HIV infection and prevent progression. However, there is no preventative measure in the way of vaccination. We do not have vaccine against HIV. And the treatment is efficient, but it only suppresses viral replication. It cannot eliminate the virus. It involves combination of drugs that target reverse transcriptase, viral reverse transcriptase, and viral proteases.